Thank you for the invitation and the opportunity to speak. I'm going to give you a brief presentation on taking a journey from data to biomarkers to surrogates to hopefully truly optimized PKD trials. And this is based on the previous work that was done at CPAT through the collaboration with the PKD Foundation and the PKD Outcomes Consortium. But I'm also going to talk about where the consortium is going in the very near future. So with that, I'm going to cover aspects of what you see in the middle here, model-informed drug development, and how we are optimizing the development of solutions to expedite not only the adoption of model-informed drug development, but more importantly, truly accelerate the development of medical products for PKD. One, for those of you that were part of the journey a few years back, we didn't necessarily have these two other components at CPAT, but now we do. And so I'm going to mention some aspects of the Rare Disease Cures Accelerator data and analytics platform and how we are going to leverage the power of that platform for the benefit of the PKD consortium, but ultimately PKD drug development and PKD patients. And I'm going to mention some aspects of also how the interaction is going to go with CPAT's quantitative medicine program for the generation of those solutions. But I'm going to start with walking you through a little bit of the history, focusing on the evidence that was generated for some of the successes that have been achieved through the collaboration of the PKD consortium. As you know, the collaborative effort of the consortium achieved the qualification by both FDA and EMA of total kidney volume as a prognostic biomarker for trial enrichment. Now, what does that mean? And so, in summary, what both decisions mean is that you can use, as a sponsor, total kidney volume at baseline as a biomarker that can enrich your population by maximizing the likelihood of, in the population you include, actually observing clinically meaningful events during the course of the trial. And what is that clinically meaningful event? Through conversations with the agency at the time, it was focused on the loss of 30% of kidney function. And that loss is represented by another biomarker, EGFR. And so, in summary, you can use total kidney volume at baseline to better select the patients that you want to enroll in a trial so that you maximize the likelihood of actually observing events of people losing 30% of their kidney function as measured by EGFR. That's great. And that was certainly a major victory. It was the first imaging biomarker to be qualified for any disease. And so that is certainly a victory of this collaboration. But what was the evidence? What was the evidence that was provided to support that decision? It all hinged on disease modeling and biomarker modeling. And so the underlying evidence that was presented to both FDA and EMA to support that decision was actually a couple models. And the first model is a model that describes total kidney volume progression. It's not just total kidney volume at baseline, but the actual dynamics of total kidney volume change over time with the relevant sources of variability. That model and parameters derived from that model drive another model which takes into account the time-varying probability of patients losing that 30% of EGFR. And what are the sources of variability? Age is certainly one. Baseline total kidney volume is another one. That's the whole point of the whole modeling exercise, but also EGFR at baseline. So if you really look into that, you'll see that it's two models, one model for total kidney volume over time, and one model for the time varying probability of individuals losing 30% of their eGFR, accounting for their eGFR at baseline. That doesn't mean that the models tell you the actual dynamic changes over time of eGFR. And what that does, just to give you an example, is this. You can take a look at the baseline age of the population you want to enrich, the baseline total kidney volume, and you can estimate over time how the probability of losing 30% of your eGFR changes. That's really what the evidence was. And the decisions that were made to go for that type of evidence were not taken in a vacuum. They were taking into account the feedback from the regulators and the contributions from the clinical experts and the drug development experts that were part of the consortium. And that's great. That really provides a useful, actionable tool to expedite drug development. Now, in further conversations, again, understanding the evidence that was provided for the qualification of total kidney volume as an enrichment biomarker, eventually the consortium, together with the commitment, support, and guidance of the FDA, reached a point where the FDA designated total kidney volume as a reasonably likely surrogate. And on the slide, you see the definition of what that means. It essentially means that you can use total kidney volume as a primary endpoint for accelerated approval discussions with the agency for PKD drug candidates in the autosomal dominant form of the disease. That's also a major win for the field, because now we not only have total kidney volume with confidence to be used as an enrichment marker to optimize patient selection, but we can use it as a primary endpoint in those longitudinal trials. That, of course, was taken up by our industry colleagues, but then they, being smart and being committed to developing efficacious and safe drugs for PKD, they started asking the right questions. And some of the questions you see on the slide. And you'll see that a lot of those questions have to do with one construct. Okay, I have a surrogate and I have a qualified enrichment marker. Tell me how I'm going to design the trials with that enrichment marker and that surrogate. And you'll see that the evidence provided for the TKV qualification does not necessarily answer all those questions that are relevant for clinical trial design. And that's okay. This is not by any means an indictment of the evidence that was presented for qualification. The evidence that was presented for qualification was the adequate evidence to reach that kind of a decision. But now we have other questions that need to be answered in order to keep moving the needle forward to solidify drug development, expeditious drug development for PKD patients. And so, considering the fact that there are certain questions that are very critical for clinical trial design that are not necessarily answered by the evidence that was generated at that time to support the qualification of TKV and its eventual designation as a surrogate, then what do? How do we help industry, and how do we help our regulatory colleagues be able to answer these questions in a truly quantitative manner? The answer is the development of a clinical trial simulation platform. And what are going to be the components? This is going to sound similar to the evidence that was presented for qualification, but it's not identical. There are some very important differences, and we're going to get through those. The first component is, yes, a longitudinal model. A longitudinal model of what? Of total kidney volume dynamics. Okay, we already have that, yes, but what we're adding to it is another model of the longitudinal dynamics of EGFR, not the time varying probability of losing a certain percentage of EGFR, but the actual true dynamic change of EGFR over time, together with total kidney volume over time. So this is a multivariate model that was not part of the original evidence to support the qualification. Once you develop that model, you can directly derive any percentage change of EGFR, whatever percentage you want to look at. Whether or not that's a clinically meaningful percentage change, that's another question, but the model allows you to derive directly any percentage change of EGFR over time, together with any change in total kidney volume over time. And then joining that multivariate model that looks at TKV and EGFR dynamics over time together with a survival model, but that doesn't look at the time varying probability of a certain percentage change of EGFR, but that looks at the time varying probability of developing end-stage renal disease, which is certainly a clinically meaningful endpoint that happens later over time. But that is important to understand for the use of TKV as a truly optimized surrogate marker. And so where are we right now? Some of our industry colleagues have taken the lead, and they have developed the first part of that clinical trial simulation platform. They have developed a multivariate model that looks at the dynamics of TKV and EGFR together, which again is a great amount of work. But that's only one component of the clinical trial simulation platform. What we are starting to do now through the PKD Consortium, but now with the added power of the RDC-ADAP platform and the Quantitative Medicine program, is take that previous great work from our industry colleagues and validate that multivariate model for TKV and EGFR dynamics. But we're going to develop that other joint model for the description of the time varying probability of developing end-stage renal disease. And we're going to take that collection of models, and we're not just going to stop at that and present that evidence to the regulators. No, we're going to take those models and we're going to package them into a comprehensive clinical trial simulation tool or platform that will allow the sponsors and the regulators to run very comprehensive simulations of different types of scenarios for different types of design options, taking into account their knowledge that they have about their therapeutic candidates. And just as we did with the evidence for the biomarker, we're going to present that to the regulators through formal pathways that both agencies have for their review and potential endorsement. In this case, not of a biomarker, but in this case, of a quantitative drug development tool. And there's the Fit for Purpose initiative, which is the FDA mechanism, and then the qualification of novel methodologies in drug development. That is the mechanism at EMA. And they are tried and tested mechanisms for the review of these kinds of solutions. So in summary, this is what our industry colleagues have developed, and that we're now initiating the validation process for this multivariate model that looks at EGFR dynamics over time and TKV dynamics over time. And so this is an advancement. This alone provides an advancement and a refinement of the original evidence that was developed back in the day to support qualification, because now it's not just about a specific percentage loss of EGFR. You can derive any percentage change of EGFR or any direct change of EGFR, just as you can model any direct change of total kidney volume over time. And what we are initiating due with the data we have in-house from the original contributions of our partners in the PKD Consortium back in the day, is the development of that parametric survival model to understand the time varying probability of developing ESRD as driven by a multivariate model that looks at the dynamic changes of eGFR and total kidney volume together. The work is only starting, but we already have a solid foundation with the data we have, and we have a solid foundation with the contributions of our members, and I'm going to explain how things are going to move in a really efficient fashion now that we have the power of our DCA-DAP and the Quantitative Medicine Program at CPATH partnering with the PKD Consortium. So with this collection of models, new models, more refined models than before, packaged into a clinical trial simulation platform, we're going to be able to answer all of the questions that our industry partners are asking since the designation of total kidney volume as a reasonably likely surrogate. What this means in reality is that the consortium is providing sponsors and regulators the quantitative machinery to really leverage the use of a reasonably likely surrogate marker. I personally think that this is going to provide a template for future efforts of really optimizing the understanding and the deployment in drug development of surrogate markers that exist now and surrogate markers that will be designated in the future. So what are we looking at in terms of the actual next steps for the effort? We're going to expand the PKD database, but now we're going to expand the database leveraging the power of the Rare Disease Cures Accelerator data and analytics platform. And I'll explain that in a second. We are also going to leverage the experience and expertise of CPATH's Quantitative Medicine Program, all in collaboration with the PKD Consortium, to complete the development of this proposed clinical trial simulation platform. But we're also, through the standing that the PKD Consortium has and the connectivity that the PKD Consortium provides for regulatory strategy, we're going to present these solutions to both FDA and EMA for their specific review as a quantitative drug development tool through the mechanisms that each agency has defined for that purpose. Now, you might be asking, what is this RDCA-DAP notion that you keep talking about? Well, here it is. And so this is an effort that was initiated through funding from the FDA to CPATH in partnership with NORD to develop a platform that would allow the integration of multiple types of data sources, not just clinical trials, not just observational studies, but registries and many other types of data sources like electronic health records, additional biomarker studies, genetic type studies from GWAS studies to genotyping studies, etc. To then be able to have a mechanism for the integration of all those types of data sources and be able to generate the supporting foundation for those data to actually make sense for then the development of sophisticated solutions. I'm going to spend a little bit more time explaining how this is added value to what the PKD Consortium did before. When we started this for the qualification of TKV, we were able to integrate data from observational studies and patient registries, which is great. I mean, in an orphan indication like PKD, that was certainly transformative. And we were able to generate this, a data warehouse, through integration of data and then standardization into a data warehouse. What RDC-ADAP provides as an added value is the expansion of the types of data that we are now able to handle, and a mechanism to handle those additional types of data that are really murky in their nature to begin with, just because that's the way they are. This is not a criticism of those types of data sources. And we are building that platform in a way that is flexible enough to handle those types of unstructured data in the form of data lakes, so that we can turn them also into data warehouses to generate these solutions. Let me spend a little bit more time clarifying those terms. A data warehouse is what you are accustomed to hearing us talk about at CPAP for the integration of the data sources we integrate, where everything, just as in a warehouse, is very defined as to where you'll find the different parts of the databases. It's very well defined where you'll find the demographics, where you'll find the biomarker information, where you'll find the outcomes information, etc. That's great, but with the vision of expanding all these types of data sources that we can integrate for the benefit of rare and orphan indications like PKD, we're going to have to handle data that are quite murky to begin with. And that is where the concept of a data lake comes into play. The term came about, and the way to understand it is like a lake, literally, where there are layers of water in that same body of water called a lake. There's different layers of water with different temperatures, different solutes, different densities. And those layers are defined, but the boundaries between those layers are not really that strict. They're fluid boundaries. It's not like at a data warehouse where everything is clearly placed and demarcated. That flexibility allows the integration of a multitude types of data sources. But from data lakes, you can generate data warehouses, and that is the power that we need to generate all types of different solutions that can expedite and help drug development for rare and orphan indications. And because we had a consortium in PKD, we made it our mission to ensure that there was connectivity from the very beginning between this platform and the PKD consortium, so that we can develop these types of actionable solutions that we've been talking about. The clinical trial simulation platform that I just talked about, to ensure the proper adoption and deployment of the reasonably likely surrogate in the form of total kidney volume, is going to be ensured by having this additional capability and added value to handle multiple data sources. And of course, the data we got contributed before is now hosted through the RDC-ADAP platform, which increases the efficiency of the effort tremendously. But back to the beginning, it's a three-way collaboration between the PKD consortium that brings in the scientific knowledge and the driving of the regulatory strategy for the whole effort. RDC-ADAP bringing the power of being able to handle multiple types of data sources and maximizing the efficiency and the utility of those data. And then the quantitative medicine program that was created in part thanks to the success of the qualification of total kidney volume and the generation of those models, all partnering together to generate these actionable solutions and continue to have impact in accelerating and facilitating drug development for PKD. So with that, I'll leave you with just one equation. Success is a function of time and people. So thank you, people, for spending the time to be here today and hearing about this exciting collaboration. With that, I'll conclude my presentation, and thank you very much.